So I guess we are ready to get started. So again, good morning, good afternoon, or good evening to all of you, depending on your time zones. It is really a great pleasure to have all us together again. It's been a while since our last gathering. So we're really very excited about having all of us together today for a topic that it is really of great interest for many of the members of the medical device community, and probably the top in the list is COFEPRIS, as there are many discussions happening, and we're really very appreciative to have had COFEPRIS really very strong involvement in the development of this program, as to make sure that the objectives that they do have and all the capacity needs in this regard are met through this webinar. So welcome everybody. And as we generally do in our sessions, again, we have this webinar format where with the session being recorded, and we will make it available through the coalition's website along with the presentations. Simultaneous interpretation is also available using the globe icon at the bottom of your screens. As usual, all attendees' microphones are closed to prevent background noise, but we are always welcoming for this to be as interactive as possible. Therefore, we invite you to post your questions using the Q&A feature that is also located at the bottom of your screens. And once every panelist or every session is completed, then our moderator is going to be helping posing the questions to the speakers. And for the speakers, thank you very much in advance for opening up your cameras while doing your presentations or while responding questions, because that makes our interpreters work much easier, and the quality of the interpretation is, of course, higher. So thank you very much for that. So with that, as you have already seen in the program, here is the agenda that we're going to be following today. So we have Catherine Serrano opening the session, and then moderating the three presentations that we have today is Besa Bunichi. And we're going to be covering fundamentals of establishing sterile barrier systems for medical devices, package stability, shelf life, and package performance by Stephen Tordo. Assessing device performance for the label shelf life, and considerations for assessing use life of reusable devices. And with these, then I would like to introduce, even though Katie is very well known by all of us, it is never too much to share with you a little bit of her bio. So Katie Serrano is the director for FDA's Latin America office. She has over 15 years of experience in the medical device field, with the last 9 years spending at FDA's Office of In Vitro Diagnostics and Radiological Health in the Center for Devices and Radiological Health. At the FDA, Katie has worked in a variety of technical, policy, leadership, and management positions, including joining the agency as part of the inaugural class of Commissioners Fellows in 2008, working as a scientific reviewer and serving as the Diabetes Diagnostic Devices Branch Chief and Deputy Director of FDA's Division of Chemistry and Toxicology Devices. She also served as the technical and policy lead for the agency's efforts related to laboratory developed tests. Ms. Serrano received a Bachelor of Science degree in Biomedical Engineer and a Bachelor of Arts degree in Spanish from the University of Minnesota Twin Cities. Before working at the Food and Drug Administration, Ms. Serrano held positions as a regulatory affairs professional at Boston Scientific Corporation and as a biomedical engineer at the United States National Institutes of Health, supporting new programs focused on cutting-edge technologies in the field of bioengineering. And before transferring the word to you, Katie, I think that now it is over 16 or 17 years of experience. I guess this bio is a little bit outdated, probably 2 years worth outdated. So sorry for that. I just realized that while reading this, but I mean, your wealth of experience just keeps increasing and increasing. So thank you very much, Katie, and the floor is yours.
No problem. Thank you so much, Sandra. And first of all, I really wanted to say thank you to the MDRC for allowing us the opportunity to provide this webinar. And also, of course, to the panelists who are participating. I think we really appreciate your time and sharing the wealth of knowledge that you have with this group of regulators and industry across our region. And of course, I want to thank everybody who's on the call with us today, because these kinds of events we put on really for you. We want to explain our approach and really have the opportunity to talk about differences in how countries in this region may approach these issues, and how we can come toward more of a consensus as a region. Stability studies and shelf life in medical devices are really critical in ensuring quality, safety, and effectiveness of these products. The studies help determine product stability under various storage conditions and provide data for establishing appropriate expiration dates. By using standards and harmonizing the criteria of how these studies are performed and actually reviewed by regulatory agencies, and how expiration dates are established, this actually helps facilitate more product to the market, because it reduces unnecessary regulatory burden and hurdles, and really makes sure that patients have access to high quality, safe, and reliable products. Also, I think coming out of the COVID pandemic, it became very clear that having a common approach to performing stability studies and reviewing stability studies by regulatory agencies really helps ensure robust supply chains, that this is a critical component in ensuring the accessibility of different products. So I think it's a very timely topic, it's a very important topic, and we really hope that you enjoy the presentations that our panelists have for you today. And we'd love to hear from you. We'd love to have you actively participate in the question and answer session at the end, and provide us feedback about what additional topics you might need more information about as we move forward. So again, thank you very much. I hope we have a very active panel today with lots of questions from the audience, and thanks again.
Thank you very much, Katie, for the welcoming remarks, and Sandra for the housekeeping messages. I'm Vesa Vunici. I'm the International Relations Specialist with the FDA Latin America office. And I'm really looking forward to moderating this particular webinar, and it is my pleasure to welcome all of you. First, as a first speaker on the panel, I would like to introduce Stephen Tortil. He's a microbiology reviewer with the Office of Orthopedic Devices at the Center of Devices and Radiological Health at FDA. He leads the center's sterility interest group, which provides continuous training in these areas for the agency. He's an active contributor to the national and international standards development organizations, and he has been FDA's primary liaison for many of these standards groups. Altogether, he has experience of 10 years being a reviewer and 25 years at the FDA. So he has an extensive knowledge in this area. So it's my pleasure to welcome Steve to discuss the first topic. But I also would say, please, to all the attendees, please take advantage of having these expertise here today to put questions on the question and answer box. So we can get to those later. We have a specific session on question and answer. So we would really like to answer those. So please feel free to put those questions on the box. This particular topic that Steve will be discussing is conducting package stability and performance for terminally sterilized single-use medical devices. And then he is also going to be discussing establishing use life for reusable medical devices. Steve, feel free to take over when you're ready.
Okay, I'll take over now. I'll share my screen. Bingo. Okay, hopefully you can see my screen now.
That looks great, Steve.
Okay. Well, thank you for joining, everybody. Good day, and thank you for your interest in this topic. I'll be presenting on two particular topics, package stability and package performance validation for terminally sterilized single-use devices. And then after that, I'm going to talk for a little bit about establishing use life or service life of reusable medical devices. And just as general context, when we do our regulatory review of devices, they fall into two categories that we see the most of: class 2, or sometimes they're called 510K devices. These are moderate risk devices. And we're going to go over in a minute some of the basically the 5 points in the sterile devices guidance document that we typically want to see documented in submissions for that. And I'll also be talking about class 3 devices, or sometimes called PMA devices. These are higher risk devices, and we comprehensively evaluate all test data upon submission there. But I do want to make an important note. There should be comprehensive completed testing for both types of devices, both general classes of devices. We just look at them at slightly different times. In both instances, all required testing must be completed and documented for each type of device. What differs is when it is reviewed, and even that may vary depending upon the device type, because some unique devices may have a device-specific guidance, and they may want to see data up front. Otherwise, it might be reviewed during a routine inspection process. So again, all the testing should be done, and all the data should be available. When we look at it, varies a little bit. So objectives for today: to understand packaging validation methods for terminally sterilized single-use medical devices. And then we break that down based on the standards, but also based on our review practices, into two categories. That's package stability, that's the aging and shelf life testing, and package performance. How does it handle simulated ship or real world shipping and handling? And how does it maintain package sterile barrier integrity during that process? And item number 2, to understand the use life, reuse life, or service life options for reusable medical devices and how these are established. I'm just highlighting the 3 different terms there. We're going to be using them, or I'll be using them interchangeably. They all mean the same thing. This is for reusable medical devices, and they don't last forever. So every time after they're reprocessed, they're subject to some sort of evaluation to make sure that they're still performing the way they should be. Then on the bottom note, product stability and performance are also evaluated. There are a lot of things that are evaluated during regulatory review, but they fall into other categories. There may be, medical device performance in itself, shelf life or service life. I'm not going to be talking about those here. I'm just going to be talking about use life from a microbiology point of view, and a little bit more for the reusable devices. So I'm going to introduce you to some terms, but the first and most important one for context is a sterile barrier system, and we often abbreviate this as the SBS. This is the minimum packaging that minimizes the risk of ingress of microorganisms and allows aseptic presentation of the sterile contents at the point of use. And from a microbiological perspective, the sterile barrier system, the SBS, should be designed to assure adequate sterilant penetration. That's going to vary depending upon the sterilant, as well as maintenance of sterility. And that's from the point of sterilization and manufacture to the time and place of the medical device use. Again, I'm emphasizing these 2 subcategories. FDA recommends package performance testing be designed and implemented to assure that the packaged product can withstand the rigors of real-world worst-case shipping and handling. And FDA recommends that package stability testing be conducted to assure that packaging will maintain product sterility for the duration of the stated expiration date. That's the shelf life on the product. And for context, I'm going to introduce you to what we call our sterile devices guidance document, published in 2016. Here's the formal title here: Submission and Review of Sterility Information and Pre-Market Notifications. That's 510K. That's the class 2 device types for devices labeled as sterile. This is a freebie, so you can download this from our website. I should have included a link here, but it's real easy to find. Just Google FDA guidance sterile devices, and it'll pop right up. This is a representation of the table of contents for that guidance document. I just want to highlight at the bottom what we typically look at during, when we're documenting during review are these 5 items down here, but the last one is packaging. So we're going to take a little bit of a closer look at Roman numeral 5 and item number 5 in particular. This is a basic introduction to that guidance. So what we typically want to see documented is sterilization method and description of it. That's 1. 2 is the validation method. So for example, maybe the half cycle method for ethylene oxide sterilization. Sterility assurance level, pyrogenicity claim, and lastly, what we're going to talk about is a description of the packaging. That's the sterile barrier system and how it maintains the device's sterility, and a description of the package test methods, but not the package test data itself. So for 510K review, we don't typically look at test data. You can submit it, but we don't typically look at that. If you submit it, we'll look at it. That's typically looked at upon inspection. Sometimes, if there is something of concern in your application and we want to look a little deeper, we may ask for the package test data on that in advance, during 510K review, but it's not typical. And as a footnote on the bottom, this is not a footnote in the guidance document, but just worthy of noting, some device submissions should provide package data. And that is for very particular device types. An example is pre-filled saline syringes. This depends on the device type. So know your particular device types and review practices. So you may want to check, with the agency to see what category your device falls into. But let's take a closer look at item number 5. Description of the packaging and the test methods. There is in that guidance document a footnote, which is really helpful for, it's basically 2 sentences, but it means a lot. FDA recommends that package test methods include simulated distribution and associated package integrity testing, as well as simulated and/or real-time aging and associated seal strength testing. So what we're talking about here are those 2 categories of performance and stability. And the rest of the note says, please refer to FDA recognized version of AAMI ANSI ISO 11607 series of consensus standards. So we're going to be referring to that 11607 a number of times in the coming slides. Taking a closer look, about those, about these standards, I'm a little bit limited in the sense that sometimes when I talk within the agency, I know that we have the standards, that everybody owns them. And I know that some of you may not have all of these standards. So I've got a little bit of a copyright issue in terms of how directly I can quote standards. What's quoted here is our guidance. That, again, that's free, but the standards, that's something that you have to acquire. So I have in some instances, paraphrased a little bit, but in other instances, I have some direct quotes that I have copyright permission to reproduce here. Okay, so this is the basic outline. Looking a little bit closer at item number 5. This is a description of the packaging and how it maintains the device's sterility and the package test methods. So in order to address this in a 510K submission, a class 2 submission, you can either just explain that you have done simulated shipping followed by package integrity testing. That's your performance testing, and that you've done aging followed by seal strength testing. That's your stability testing, or a statement of conformity to 11607 series of consensus standards, because 11607 is an FDA recognized consensus standard. So we have participated in it, we've recognized it, and it's available for all our reviewers and all of you to use as a great reference, a great standard. So looking a little bit closer at number 5, another example, okay, the example I just showed, these are the simpler things to do, but you can also, this is also a very acceptable answer for a more comprehensive response. For performance testing, you can spell out the standards that were used. So there's simulated distribution. ASTM D4169 is very popular, very commonly used. And then once that shipping is simulated, then a package integrity test, such as dye penetration, that's also very commonly used. And for stability, simulated aging in ASTM F1980, That's the number of the standard, not the data was published. Followed by seal strength testing. That's ASTM F88. There are other permutations. I'll let you know that in advance. In fact, I'm going to switch to a slide to show you how many permutations there can be. But if you're familiar with other processes such as sterilization, typically you're looking for a straightforward test with a simple endpoint. For example, if you're doing a sterility test, if you're validating a sterilization process, you're going to have biological indicators in there perhaps, and you want to see that they've all been inactivated, that they're all dead. That's a simple test design and a simple acceptance criteria. When it comes to packaging, it can be much more complicated. And this is an example of how complicated it can become. It says all over it, example, and it's a hypothetical and it's draft because there are so many variances that can occur. There's so many different ways that you can test your packaging, but this is a common one that we've seen pretty frequently. I'm going to go through some of the finer detail of the text for you. In the intro, this represents a hypothetical series of simulations and subsequent tests intended to provide a high level of assurance that the packaging will demonstrate adequate stability and the packaged product will be able to withstand real-world worst-case shipping and handling without package failure or sterile barrier breach. The details of this test schedule may or may not be appropriate for other products, as test procedures should be developed on a case-by-case basis. In general, the agency considers breach of sterile barrier system to more likely be event-related than time-related. And that's a line that's out of the 11607 series. That sterile barrier system is more likely, breach of sterile barrier system is more likely to be event-related than time-related. In other words, if it comes out of the industrial sterilization chamber and you put it on a desk across the room and let it sit there for 5 or 10 years, it's a lot more likely to maintain its sterility than if you were to take it out of that sterilization chamber and ship it around the world onto trucks and planes and altitude changes and things like that, and have it and have the product evaluated 4 or 5 days later. There's more things that can happen to it. Anyway, I'm going to go through this, but I'm going to go through it a couple times so that we can get a better idea of what's really going on in the validation process. Typically on the far left corner, there's like a sealer pilot study, and that helps establish the critical cycle parameters there, the cycle parameters, that is temperature, dwell time, pressure, in order to establish the optimal sealer settings. In addition to that, okay, these are just references again to the standards. But then the two big tests that we typically look for are right in the middle 2 columns there. Stability, we're looking for, this is just an example. If somebody wants a 3-year shelf life claim, they can do a 3-year accelerated aging simulation. 30 packages is recommended. I'll get into why shortly. And then that's followed by peel strength testing. So that at the same time, we do accept this as a temporary evidence, but at the same time, you should be starting real-time aging. This is confirmatory testing, and it should also be followed by the same test type, peel strength, typically performed. Again, there are other possibilities when it comes to the test type, but the peel strength is what we see the most of. The second category, and you can see these are both in big bold text because they're the real important ones. Performance, and this is for shipping and handling, simulated shipping and handling, or what's also called distribution. And most frequently, we see the ASTM D4169. Distribution, this is an example. Again, distribution cycle 13, assurance level 1, 30 packages, and included in there is handling, simulated vehicle stacking, loose load vibration, low pressure, vehicle vibration, and there are several others. I haven't included them all here. I'm a little bit leery again of the copyright infringement, but I'm just showing you, what we typically do get. And that can be followed by validated package integrity tests, such as die penetration. and the standards provided here. There's also, in the very last column on the right, there should be time zero peel strength testing, because the second column that you're looking at, the stability test, that's giving you a shelf life to make sure that it can maintain product sterility for, in this case, 3 years. We also want to know, what if the product shipped immediately and used immediately? We want to know that that seal is adequate. So we also want to know that you're doing time zero peel strength test. Again, 30 samples is ideal. 2 notes in the bottom right-hand corner. These are valuable. If packaging is comprised of a double sterile barrier system, both internal and external packaging systems should be tested. So if you've got a double sterile barrier system, you can't just test one of them. You have to test them both. And if the testing is not done on both, if it's conducted just on one, then special labeling has to go on there to indicate to the end user that only one of the sterile barriers has been, only one of the barrier systems is validated to be a sterile barrier system. And at the bottom, these are references to FDA's recognized consensus database. So I'm going to later on, I'm going to switch to this link and show you what that is. But this is publicly available listing of all FDA recognized standards, and as well as additional guidance that can be is cited for recognized. Okay, there's recognized standards, and then there is relevant guidance. So that website will help you identify all of that information. And there are examples down here. I just checked this a couple of weeks ago. All of these standards listed at the bottom, the 11607, parts 1 and 2, ASTM D4169, the accelerated aging, the dye penetration, seal strength, and visual inspection, they're all FDA recognized when I check them. So keep in mind, you need to check these, stay current on these, check the website, because occasionally the standard will change. Sometimes it'll become outdated. Sometimes we don't recognize all standards. We just, that's just the nature of the game. And again, double asterisks there. All test methods should be validated, use statistically significant sample sizes, and we recommend 95% confidence, 95% reliability. That will give you the 30 test packages here and the 60 test packages here, and include predetermined, scientifically justified test endpoints. So these fall into statistically, these fall into 2 different categories for test types. That's why there's different sample sizes, 30 and 60. 60 is attribute testing, and 30 is for variable data testing. And just as an introduction, this is 11607 part 1, has to do with materials for sterile barrier and packaging systems, sterile barrier systems. And part 2 has to do with validation requirements for forming, sealing, and assembly processes for sterile barrier systems. We'll take a little closer look at number 1, and you can see I've included here because I'm legally allowed to do this. This is the scope, and it does not constitute copyright infringement if I share this with you, but you can see here requirements and test methods for materials, preformed sterile barrier systems, and packaging systems that are intended to maintain sterility of terminally sterilized medical devices until the point of use. And here's some additional information as to what it does and doesn't cover. And this is part 1, 11607 part 1, clause 1, which is the scope. And if we switch to 11607 part 2, this is also clause 1, which is the scope, but it spells out requirements for the development and validation of processes for packaging medical devices that are terminally sterilized. These processes include forming, sealing, and assembly of preformed sterile barrier systems, and packaging systems. It applies to industry as well as healthcare facilities. Going to do a little quick introduction to terminology. Aseptic presentation is a transfer of sterile contents from its sterile barrier system using conditions and procedures that minimize the risk of microbial contamination. The microbial barrier is a property of a sterile barrier system to minimize the risk of ingress of microorganisms. packaging system is a combination of the sterile barrier system, the SBS, and protective packaging, which is two definitions below. So the protective packaging system is a little different. Preformed sterile barrier systems, it's a sterile barrier system that is supplied partially assembled already for filling and final closure or seal. There's an example given there, and you can see some manufacturers will buy a preformed sterile barrier system, and it'll include, for example, the seal A and seal B and the chevron seal. C is what the manufacturer will put it in. So in other words, they'll put a device in here, and then will seal it at the site of manufacture themselves. That's what a preformed sterile barrier system is. Protective packaging is the material configuration of materials designed to prevent damage to the sterile barrier system and its contents from the time of their assembly until the point of use. So this can typically be a shipper, corrugated, cardboard kind of thing. It's nothing that constitutes the sterile barrier system itself. That's the sterile barrier system that we talked about earlier. And process parameter, these are the specifications for the process, what I mentioned earlier, the pressure, the time of the sealing process, the pressure, and for those that are heat sealed, the temperatures used. Seal integrity, characteristics of the seal to minimize the ingress of microorganisms. Seal strength, mechanical capacity of the seal to withstand force. And here we go again, sterile barrier system, the minimum package that minimizes the risk of ingress of microorganisms and allows aseptic presentation. And terminal sterilization, process whereby the product is sterilized within its sterile barrier system. If you can think of some other devices you're probably familiar with, like GI endoscopes, are typically high-level disinfected. They're not in packaging. They're in a high-level disinfection machine, an automated endoscope reprocessor. That does not constitute terminal sterilization. It has to be within its final sterile barrier system to be called terminal sterilization. So coming back to this, we're going to look at it a couple of times more, just to give you an idea of what's, of the overall context. And I wanted you to know about one additional other, hang on a second, I'm getting ahead of myself here. Oh yeah, so we're going to step right into the stability. We're going to take a look at stability and go through the details of that, and then we're going to come back and look at performance. So let's take a look at stability. We're going to actually consider both parts, part one and part two, and there's a third, guidance, which we'll talk about in a minute. But I just want to point out to you, this is 11607 part one as part of Annex B. And again, I've gotten permission I've copyright permission to reproduce this here. This is part of a 10-page list of standards that are cited in this 11607. Many of these are ASTM standards, but there's a mix of other international standards also. Everything that appears here is not necessarily an FDA recognized standard. So again, you have to turn to our database just to make sure. And what I'm going to do is I'm going to focus in on the left page, and we're going to look a little closer just to give you an example of what's in here. And because we're going to be talking about accelerated aging now, this gives you an introduction to the annex, but also gives you an introduction to each particular standard and some of the details and relevance of it. So this is accelerated aging, ASTM F 1980. I'm going to move on and also talk about, this is that third ISO standard, 16775. And I see a typo. This is for steam sterilization. I apologize for that. In the top left corner, it says 17665. That is steam sterilization. You can see the numbers are very, very similar. The actual standard is 16775 for packaging, and it's entitled guidance on the application of 11607, parts 1 and parts 2. And we're going to take a little bit of a closer look at that just as an introduction. And that is, there's basically 4 pages of tables of contents. And this is one version. I think this is about to be replaced with an updated version, but this is just to give you an idea of what's in it now, what's been in it in the past, and what'll be in the future. But you can see I've highlighted a couple lines: general performance requirements for materials, 5.6 additional requirements for sterile barrier systems and preformed sterile barrier systems. So this is all additional guidance. 5.9 is compatibility with sterilization process. 5.18, packaging families. And we're going to switch to the next page and talk a little bit more about packaging families. Here we go. 6.4, process validation of packaging families. Just a digression, a note on that. For every process that we evaluate, and it can be ethylene oxide sterilization, or it can be radiation sterilization, it can be anything. Very often, a master product can be identified, and then other products which are very, very similar, I would say very, very, very similar, can be adopted into that process. That's quite common, and it saves manufacturers from validating each and every product if you can identify a master product. But every process is different. So how you would create a family for packaging is different from how you would establish a package, family for ethylene oxide. Ethylene oxide, you would have to turn to 11135 and TIR 28 in particular for establishing a family. Radiation, same thing, but there are different rationales. So you can't establish a family for packaging and assume that it applies to radiation, which calls for its own clause within its own guidance, within its own standard. But again, it's designed to make things a little bit, a lot easier to minimize, to make it less burdensome on the manufacturer. So for example, if you're making a medical device that's 10 inches long, and you've got different versions of it that are half an inch shorter by half-inch increments, then if you do the master, the larger one, you should be able to justify. This is just an example, and it's very rudimentary. But you should be able to come up with a rationale based on the guidance in here for establishing appropriately a packaging family. Process changes and revalidation are also mentioned, and towards the end in the annexes, guidance on establishing process parameters, guidance for industry, sterilization considerations, guidance for industry and healthcare facilities, and investigating failures. It's loaded with great information, so I just wanted to make sure you know that it's available. 16775, not the other number. Getting back to item number 5, examples of packaging. You may already know this, but it should be allowing for sterilant penetration and sterility maintenance. These are examples for ethylene oxide. Should be breathable materials. Tyvek Mylar is a good examples. Tyvek PETG. For steam, paper plastic is common. Possibly for low temperatures, Tyvek. But only for low temperatures, and you really have to validate. Of course, you have to validate it, but you have to be real careful for Tyvek. If you use temperatures that are too high for Tyvek, what you wind up getting is very expensive shrink wrap on your product. You will melt the Tyvek. Radiation can be non-breathable, foil, mylar, film, dry heat. That varies. In my experience, I haven't seen this a lot for the products I review, but dry heat, there are different types of packaging for dry heat. A question that came up. Typically, do the labelings or the IFU need to be part of the sterile barrier system stability testing? So if they can interact, anything that can interact with a package, and cause package failures, that should be tested. So if you've got if you've got a double sterile barrier system, you should have the product inside the package during performance testing, shipping and handling, but you should also have everything else that's going to be in there, because you don't know how it's going to interact with a package. And I've only seen a few failures in my experience. I think sometimes when failures occur, the manufacturer starts from zero, they start all over again, and they redesign the package. But the failures I've seen have been based on interaction of some component with the package, causing breach of sterile barrier system, or something almost as bad, something that's worse than a cosmetic change. So it's important to, let me put it this way. This is a mantra that we hear a lot in the agency. Final finished product. That's what you want to test always. Final finished product. And that applies to so many different test types, from biocompatibility, engineering, everything. And it applies to packaging. So you want to know exactly what is going to be shipping. And that's the thing you want to make sure has been adequately tested. Whoops. This is a quick high resolution scanning electron micrograph of Tyvek in particular, or sometimes what we call a non-woven fabric. And you might think that, okay, this is great. It's going to prevent, these holes are too small for microbes to fit through, and that's what keeps out the bad guys, the bacteria and other things. But actually, you'd be wrong, because even higher scanning electron micrograph shows that these are bacteria and they can fit through the holes. The catch is the way they design this packaging for something that's going to allow gas penetration is that it's so thick that it creates a tortuous pathway, and microbes don't go through it. I mean, if you turned on a extremely high pressure water hose or something like that, you could probably get them to go through. But after decades of testing, typically microbes, the tortuous pathway that's created by this sort of material does not allow microbes to go through. And here's another little thing. I've never seen data on this, but Tyvek, if you've ever handled it yourself, you might notice that it has a slight electrostatic charge. And, like when you rub a balloon against wool and have it stick to the ceiling or something like that, same thing, that it's believed that microbes stick to the outer layer of this just because of the electrostatic attraction, and they don't go in any further. I've never seen data supporting that, but it seems to be a possibility. Here is just some information again on Tyvek. The FDA does not endorse any one product or one company over another, but this is very commonly used, and I just wanted to share with you. This is publicly available information online. Taking a little bit closer look at some of the qualities, you can see that it's typically tested for tensile strength, elongation, tear strength, burst puncture resistance, things like that. That's some of the materials, quality of the materials that are used. So these are for ASTM standards. Again, these are the ones that I have listed in there. They're very commonly cited and used. The first two are for simulation, ASTM D4169. That's simulation, and the second one is for accelerated aging. And these are the test methods for afterwards: dye penetration and seal strength. And you can see how they would fit into here for stability, and you can see how they fit in here for package performance testing. Let's talk now, since we've kind of done a general introduction, more than a general introduction, I want to look at accelerated aging carefully. These are references. You feel free to reach out and take a look at it. This is the standard itself, ASTM for accelerated aging. It's ASTM F1980, but this is the year of publication. So this is the most current version, and we happen to recognize that. This is a little dated, but it's our guidance document, has a lot of good information in it. This is from an online publication. And if you go to this link, you will actually find some good information that is in the very first standard that's mentioned up there. So that's a very good introduction to the actual standard. And choosing some information from those references. This is from our guidance document and also from another company's general guidance online. As a rule of thumb, for every 10-degree increase for the tested temperature above normal storage temperature will increase the expiration date by a factor of 2. Okay, we're talking about accelerated aging. The Arrhenius equation is what's behind this. Sometimes you may hear it referred to as Van 't Hoff's, and he's the guy who trained Arrhenius, basically, or Arrhenius learned from him. The Arrhenius equation indicates that for every 10 degree C increase in temperature, you get a double of the chemical reaction rates. And this is known as a Q10 of a factor of 2. Some materials have higher or lower reaction rates, Q10 factors. So that's important to know. This is the most popular and conservative method of calculating accelerated aging. This is what we see all the time. This is again from our guidance document. Accelerated studies combined with some other additional information may be used to support tentative expiration dates, provided full shelf life studies are not available and are being conducted. That's why in that table, when I have the stability, I have 2 columns for stability. The first one is accelerated. The other one, that's the first 30 packages. The other 30 packages are for real-time aging and subsequent testing. And this is from our FDA guidance document. You can also, that's what I provided the link for. Test procedure for testing shelf life. The relevant items are number 5 and 6. Written procedure for establishing and monitoring shelf life of medical devices should include the following: accelerated aging parameters. So that's basically, the conditions, the temperature and time. And so it's going to give you the calculations, including information that validates the accelerated system. Results need to be supported by real-time testing. That's the second column. And there we go. This is a direct quote from 11607. Stability testing using accelerated aging protocols shall be regarded as sufficient evidence to claim expiry dates until data from real-time aging studies are available. And finally, an important addition here is ASTM F1980 standard suggests using accelerated aging temperature below 60 degrees. Medical devices are often engineered with delicate materials that may drastically change when exposed to temperatures exceeding 60 degrees. Here's the link for this. And in general, all materials have different Q10 factors, but most of those used in packaging have a Q10 of equal to 2. This is a figure that's actually fairly commonly found, but it is in ASTM F1980. Gives you a good introduction. Accelerated aging of polymers, time versus temperatures in this flow chart, in this diagram, show the time equivalent to 1 year of room temperature aging when polymer is heat treated at a selected temperature. And to take a little bit of a closer look, you can see, you can figure this out fairly easily. But if you look at room temperature, which is in the bottom left corner, room temperature, how long is it? What's the equivalent in weeks for a 1 year real time? And you can see, at 22, it is 52 weeks. If you scroll over, and I'm going to switch to 55 degrees, that's the most common temperature that we see accelerated aging studies performed at. You can see that going up to the middle line in here, which is Q10 of 2, that dotted line, right at 55 weeks, it drops off right about, at 55 degrees, it drops off right at about 5 weeks, which is maybe a little bit above 5 weeks right here. And you can see what I'm going to introduce you to is the calculations themselves. Accelerated aging time. This is basically the equation that you can find basically in college physics books. For 365 days of Q10 of 2, if you take this number here, which is the accelerated aging temperature, if you're going to accelerate it at 55 degrees, but your standard temperature, room temperature, storage temperature is 23 degrees. Divide that by 10. So right now you're getting up to 32 degrees. When you subtract this from this, divide it by 10, you get a 3.2. You take that Q10 factor to the power of 3.2 and you get 9.19. Divide 365 days by 9.19 and you've got 39.72 days. Accelerated aging equals 1 year of real-time aging. And this is pretty close to the most common thing we see, again, 55 degrees for accelerated aging. And we see days in here from 37 up to 41 degrees, depending upon what is established or what is assumed to be room temperature, normal storage temperature. But I'm going to give you two quick examples of, something a little higher and something a little lower. I've changed the color there, 50 degrees. If it's done a little bit colder, you can do the math. 50 minus 23 is 27, divided by 10, 2.7, divide 360, 2 to the 2.7 power is 6.5, divide 365 by that, and then your accelerated aging time is going to be 56 degrees. It's going to take a little bit longer to be 1 time of real time, 1 year of real time. And if you go up to that 60 degrees, and I've seen somebody do this once and they did have problems because they had a materials compatibility problem with that particular temperature. 60 minus 23 gives you 37, divided by 10 is 3.7, 2 to the 3.7 power is 12.99, divide 365. So that helps you do it a little quicker, but it's a delicate balance. You're going to get advantages and disadvantages however you approach this. Again, this is the standard we were just talking about, ASTM F1980. What we're going to consider now is for testing seal strength. This is the bottom one, ASTM F88 or F88M. And seal strength, whatever you choose to be your final acceptance criteria for seal strength, should be scientifically justified. There is no one single standardized acceptance criteria in a standard anywhere that is FDA recognized. There's one ASTM standard that does specify one, but we don't recognize that. It's not FDA recognized. And as long as I've been doing this, I can go back, I think I went back, my first standards meeting goes back almost 20 years ago, and I approached the co-chair and I said, what is an acceptance criteria for seal strength? And he says, we've never been able to get consensus opinion on that. A very common value is 1 pound per inch. It's very common. And we see values all the time between 1.5 and 2 pounds per inch. Very common. If you go above this, if it's too strong, it's going to be problematic because it really is going to take a lot of force. And whoever's trying to open that package is going to be pulling on the package and the product can go flying across the room. You don't want that. If it's too weak, that's also going to be problematic. You're going to have a shaky package. It's not going to be holding together very well. Values below 1 pound per inch may be acceptable, and we do see this quite commonly for smaller, lighter mass devices if scientifically justified. So if you have a very light, small product, packaging it in a, designing a package with a seal strength lower than 1 pound is okay, but again, has to be scientifically justified. That's at the end. We're getting to the end of this here. And let me think, where was I? Oh, yeah, I just want to add here, what we're going to move on to is, this is our shelf life guidance document again, where we talked about was accelerated aging, but item number 6, simulation of shipping and handling stresses plan. This is what should be provided and documented. Include vibration tests, temperature extremes, actual shipping, and intentionally mishandling the device to determine the effect of unusual circumstances. So this is for your reference, since I can't directly quote parts of 11607, we incorporated into this is our boilerplate deficiency. It's large because it has a lot of guidance in it. Sometimes we don't use the whole thing, of course. If we just need one thing, we'll ask for that one thing. But when it comes to a class 3 device, we might want to see a lot more, or we just need to, if there's nothing provided in a submission, we may need to give the submitter a lot of guidance. So, all of this text here has to do with what we've already covered: performance testing and stability testing. That's what the first bullet is about. We talk about distribution cycles and assurance level. Here's the link again to FDA recognized standard. And if you actually get this standard, I mentioned sterility, I mentioned assurance level, and that is defined within the standard, and also mentioned distribution cycle. That's also mentioned in the standards. Like I said in the beginning, there's so many different ways to do this. It can be a challenge. Actual product, this is more guidance. Actual product or simulated product should be included in packages that undergo performance testing, because that partly answers the question that came up online, texted earlier, is we want to know how the product interacts with the package. So you should have packaged product for performance testing. And for simulated aging or real-time aging, you don't necessarily have to have a product in the package. It depends on the package design. It could be justified not to have that in there, because depending on the design of the package, there may, interaction of the product with a package is very, very minimum. And it's a little bit detailed, but we can talk about that later. But it may not have to have, for aging, real time, and accelerated, it may not have to have product in the package. And the last bullet here talks about 95% confidence and 95% reliability. That's why we chose the sample sizes of 30, 60, and 30 for attribute and variable data. And for double sterile barrier systems, we want to see both systems tested. This is a little bit better introduction to ASTM 4169D. These are the distribution cycles. They go down to, I think they're up to 18 different distribution cycles now. And these are the components of each distribution cycle and the order that they should be performed. And I'm dropping in on the next slide, an example. 13 is what we see the most. Distribution cycle 13, assurance level 1. That's what we see the most of. It's not the only possible combination. Whatever is done should be validated and justified. Other DC distribution cycle 13 details are omitted because I didn't want to, again, copyright issues. This is part of the draft efficiency again. This is general guidance, but what we may or may not ask you, depending upon what you've
Lo que le
provided.
podamos pedir o no, dependiendo de cuánto nos hayan provisto.
What we may ask you, depending upon what you have or have not provided, is clarify the proposed shelf life, because sometimes it's not stated. Identify the standards. For a 510K, we may ask that more information be provided on the chronology and the test types. For a PMA, we may ask for a flow chart. Please show us, something like I've been showing you, that flow chart. How everything was done, what was done, and in what order, what chronology. And we may ask for, in that last instance, we may ask for protocols and some test reports for, depending on the circumstances. Anyway, that covers package stability. I've got a few slides. I'm going to talk about service life or reuse life or use life of reusable medical devices. So this is relatively straightforward. I'm pointing to another free FDA guidance document. This is for reprocessing. It was published in 2015, but updated in 2017. So that's what you want to look for. Reprocessing medical devices in healthcare facilities. So this has to do with cleaning and sterilization. That's not really the subject of what we're talking about today, but what is the use life of the product. So what we're going to look at is only criterion 5, reprocessing instructions should be comprehensive. And when we take a close look at that, Comprehensive instructions enable the user to understand precisely how to implement the entire reprocessing procedure safely and effectively. There may be several acceptable formats for instructions. To ensure the processing instructions are comprehensive, they should include all of the elements below. If any element is not applicable to your device, then you should state this in your pre-market submission and provide a justification. So I'm going to be jumping just to criterion number 5, item L, reuse life. The labeling should either inform the user how many times the device can be reused based on testing. All devices are different, but some devices, and we've seen these come in, we've seen all of these different categories that I'm going to share. We've seen devices that have been tested out to a certain number of uses. And in this case, the last one I saw was a robotic device. And they said, we want this device can only be used 10 times. That means you can use it, clean it, sterilize it, use it again. You can only do that 10 times because there are performance issues that occur after that. So that, actually the testing that's done on that has a very strong margin of safety. So when they tested for 10 time uses, they actually tested out far more than 10 times to establish a solid margin of safety. Provide the user with a mechanism or method to ascertain the device has exceeded its use life. In the latter case, the labeling should identify a method to establish that the device is still within performance specifications, as well as instructions for appropriate disposal of devices that are not. And examples of these are labeling that refers to a device design feature, such as a built-in automatic pre-check function. It could be, an electronic feature that is built in that you can, there's so many different device types. And I work in orthopedics, so I don't see this very often, but it can be, a very highly functional device. And there needs to be a pre-check to make sure that the performance is up to where it should be. Labeling provides performance tests that should be passed prior to use. And this could be as simple as pull the trigger on the handle and make sure that the jaws close at the other end. Or it could be more complicated than that, but this is an example. And lastly, labeling that recommends visual inspection along with acceptance criteria or failure criteria. Unacceptable deterioration such as corrosion, discoloration, pitting, cracking seals, cracked seals. I heard somebody in the user community say that they have a mantra that pitting is the beginning of the end of the use life of a product. So as soon as they see pitting, they know to dispose of the device. So how and why is this done? I want to show you again, this is the last bullet, labeling that recommends visual inspection along with acceptance or failure criteria. The devices I work with the most are orthopedics. There are a lot of metal devices and they just wear through with time. They're corroded by the different chemicals that are used for reprocessing, maybe even the sterilant. So this is another high magnification scanning electron micrograph of that type of corrosion. And you can see on the surface, this is going to be problematic for 2 reasons. One, from a microbiologist point of view, is you're going to start getting contaminants building up in here. And it could be soil and it could be viable microorganisms, bacteria, and it could be both. And you could wind up getting biofilm buildup in here. And that's going to be even more difficult to clean, or if not impossible to clean and sterilize. And that becomes a problem from a microbiology perspective. And one of our mantras, again, for the reviews we conduct is if you can't clean it, you can't sterilize it. So in other words, if you have a problem feature like this and you get soil buildup in here and you get microbes in here, that soil may shield the microbes from the sterilant, or it may, depending upon the sterilant, it may actually directly inactivate the sterilant. So things can go wrong from a microbiology point of view, but from an engineering and performance point of view, the performance of this device, this is the beginning of the end. You know, you're going to start having problems. So we want to see in the labeling some specification that addresses one or all of these. Not all, I'm sorry, I take that back. At least one of these that addresses everything, and maybe a combination of them. But what I see, I'm just giving you my examples working in orthopedic devices. They're, you know, high, kind of a lot of metallic devices. We very often see do a visual inspection, not separate from the cleaning inspection, but do a visual inspection that identifies corrosion, discoloration, pitting, cracked seals. And when you see that, dispose of the device, appropriately dispose of the device. And the very end of this is, whichever method is chosen, labeling should recommend how to evaluate deterioration in difficult to see areas of complex devices, especially those with lumens. Reuse life may also be addressed by validating the number of times the product can be reprocessed and reused, and providing this specification in the labeling. So we already did cover that. Labeling should also describe specific tracking method for the number of reuse cycles. So for that robotic arm that has 10 use lives on it, 10 use cycle lives on it, they provide a method for the end user to track the number of times, so 10 is not exceeded. So that's all I have on stability for medical devices. I'm just focusing on reusable devices, and I'm going to turn it over in a minute. This is contact information. If you have questions, like to know more, questions about what's been provided, or like to know more information, this is all of our contact information. Please feel free to reach out to us, and we'll make sure if we don't have the answer at hand, we'll find the right person who does. And that is all I have to present. And I'm happy to turn it over now to Limon. Are you there? And let me
Thank you, Steve.
stop sharing the screen somehow. Oh, there it is. Okay. Bingo, all yours.
Thank you very much, Steve. I thought that was a really good overview of how FDA conducts reviews regarding package performance stability. Thank you for sharing your expertise. And I think there's some questions that came in, but we will hold those at the end when we have that session for question and answers. But I will just take an opportunity again to just remind everyone that have joined late, or just a quick reminder that we do have that question and answer box. Please free to include questions in Spanish or English, whatever your language is, and we will try to get through those at the end after the presentations. So next, it is my pleasure to welcome Limon Sun. She's currently an assistant director within the Office of Orthopedic Devices at FDA. She joined FDA in 2010 and served as lead reviewer and master reviewer for both pre-market and post-market submissions of joint arthroplasty and fracture fixation devices. She's also an expert reviewer for materials, implant coatings, and additive manufacturing technology. And she has led or co-led the development of a number of technical guidance documents and collaborated with internal and external experts on research, publications, and standards development for the past 12 and a half years. With that, welcome Limon. Welcoming Limon here to discuss the evaluation of device performance for the labeled shelf life. So we had Steve discuss package performance, and Limon will be focused on discussing device performance for the labeled shelf life. Limon, welcome, and the floor is all yours. Feel free to share your presentation and start when you're ready.
Thank you, Risa, for the nice introduction. Can you see my slides?
Yes.
Okay, so thank you. Yeah, so good morning or good afternoon, everyone. My name is Limon Sun, as Risa just introduced. I'm also working for the Office of Orthopedic Device. So my presentation So Steve just talked about the packaging integrity, and my presentation will be focused on the evaluation of device performance for lab shelf life. So as device performance testing is very device specific, so you will see I will giving some examples basically from orthopedic area that in my slides. And here is the outline of my presentation. I will first talk about shelf life and stability of a medical device. And then I will talk about product shelf life testing. So which is basically the evaluation of the device performance for their labeled shelf life. And also I will give some, provide some recommendations for product shelf life testing. Basically examples from, as I said, mainly from OHT6 orthopedic device space, and we'll briefly touch other device types. So I do have a few disclaimers. And firstly, the language from FDA guidance documents has been accepted or summarized to fit the purpose of this presentation. For complete information, please see the cited FDA guidance documents, which are listed at the end of this presentation. And our recommendations in this presentation are based on our current thinking on a particular subject, which may be subjected to change. So also images have been obtained from online sources and are intended for illustration only. So first, I will talk about shelf life and stability of a medical device. So the information in this section is mainly summarized from our FDA guidance document called Shelf Life of a Medical Device, which was issued in 1991. While this guidance is over 30 years old, the terminology and background information are still applicable and will be very helpful for this presentation, to understand this presentation. So first, about some terminology. Shelf life is the term or period during which a medical device remains stable and suitable for the intended use. Expiration date, which is also showing using this symbol in the product labels, is the termination of shelf life, after which a percentage of the medical device may no longer function as intended. And not all medical devices need to have a shelf life. This depends if a medical device is susceptible to degradation that would lead to functional failure and the level of risk that the failure would present. Also, a medical device's shelf life should not be confused with its useful life, which is the duration of actual use or the number and the duration of repeat uses before some change results in the device inability to achieve its intended function. So the United States Pharmacopoeia defines stability as the extent to which a product retains within specific limits and throughout its period of storage and use the same properties and characteristics that it possessed at the time of manufacture. There is no one exhaustive set of criteria that would apply equally to all medical devices. USP does provide 5 sets of criteria for acceptable levels of stability, which is for drug products. And these are chemical, physical, microbiological, therapeutical, toxicological, and although this set of criteria applies specifically to the evaluation of drug product stability, it is useful as a starting point for developing criteria to evaluate the stability of a medical device. Please note that we do add biocompatibility for the medical device. And here, for each of these, here are some factors to consider for each stability criteria. So there are, for each criteria, there are numerous factors. And so some are listed just as examples. And Steve earlier talked about interactions. Here for chemical, I just want to show there are some interactions of ingredients or components or between device and packaging. So I think earlier we have a question about this. So also for the physical, I listed something like appearance. So in some cases, a significant change in appearance may cause concern to the users, even though the performance of the device is not affected. So just want to mention that all these factors are device specific. And to determine if and what shelf life testing is needed, it is recommended to perform a risk analysis. And this risk analysis should be in accordance with the currently FDA recognized version of ISO 14971 for all stability factors that we mentioned earlier to determine if and what shelf life testing is needed. So here's the current version of this standard. And this risk assessment should be specific to the medical device type with consideration to the nature of this device and its intended use, and materials and components used to manufacture the device, and the manufacture method and the process, including sterilization, as well as packaging, storage, and the transportation condition. So next, I will talk about product shelf life testing. So this information is from the guidance document, shelf life of medical device, as well as various device-specific FDA guidance documents or our current thinking. So shelf life testing is conducted to support the proposed expiration date through two types of testing. The first one is evaluation of the packaging integrity for maintaining the device sterility, basically for the device provided sterile by the manufacturers. So this is called a package shelf life testing or stability testing. This is already covered by Steve's presentation. And the second type of testing is the evaluation of any change to device performance or functionality. So this is the product shelf life or stability testing and the topic for my current presentation. The expiration date in the labeling should be in accordance with both test results, if applicable. Basically, it's the minimum value of these two results. For example, if the package shelf life is longer than the product shelf life, then on product labels, the product shelf life should be labeled. On the other hand, the package shelf life should be labeled. So just a note, in the second case, unexpired sterile packaged device may be repackaged and re-sterilized with a reworking procedure if a validated method has been provided. So here are some general considerations for establishing product shelf life, as the intended use of the device will greatly affect the tolerance level of shelf life or other failure of the device. So we recommend define a set of specifications or criteria for the final device's critical characteristics and performance with tolerance based on the intended use. Also, identify raw materials, components, packaging, and process factors to determine if materials and components have their individual shelf life characteristics in addition to their effect on final device shelf life. Also develop appropriate final device sampling plan as well as a sample storage plan. Develop a simulation of shipping and handling stress plan to determine the effect of unusual circumstances. Also establish appropriate accelerated aging parameters when appropriate. For some devices that are resolvable, their sensitivity to moisture and temperature should be addressed by packaging description and testing. Here are some general recommendations for product shelf life testing. This testing should evaluate all critical characteristics and performance of aged device samples to ensure they are within the established specifications or criteria. So again, this critical characteristics and performance are device specific, which will be further discussed using a sample or examples. And also accelerated studies are acceptable for some products to support tentative dates and storage condition as long as they are followed and supported by real-time studies. For accelerated aging, we recommend using the currently FDA recognized version of ASTM F1980 and specifying the environmental parameters established to attend the expiration date. So Steve has talked a lot about this standard earlier in his presentation. And also specifying the way in which the devices were aged and provide a rationale to explain how the results of the shelf life testing are representative of the results if the device were aged in real time. Also note that accelerated studies may not be appropriate for some products because of their complexity or their degradative nature, or lack of adequate methodology, or there is no sufficient historical data available for that type of device. In this case, the shelf life should be based on the real-time aged sample test only, which is you can put a short initial shelf life, which will be extended after additional test results. And Steve briefly mentioned the different type of submissions to FDA, specifically the 510K and PMA, 510K for class 2 device and PMA generally for class 3 device. So I will mainly talk about, mainly discuss FDA's general regulatory requirements for reporting shelf life for these two types of pre-market submissions. For 510K, the shelf life of the device should correspond to the duration of aging completed. And a summary of this test report should be submitted prior to 510K clearance. We don't need to review the full test report. And changes in the device expiration date do not require a new 510K. However, if you are using methods or protocols not described in the original 510K to support new packaging integrity or shelf life claims, a new 510K may be necessary. And for certain devices or components, testing should be conducted on real-time aged samples to confirm the results of the accelerated aging study. So this testing should be conducted in parallel with 510K review and clearance. The results can just be documented to file in the design history file. There's no need to submit a new 510K. For PMA device, the approval order should include the shelf life of the device, and test reports, not only summary, should be submitted prior to FDA approval. If FDA has previously reviewed and accepted a protocol for changes to the expiration date, and the testing was performed in accordance with that protocol, then the change to the expiration date can be made and reported in a PMA annual report. However, if a new protocol is used, a PMA supplement should be submitted for FDA approval before the change can be made. So next, I will talk about some examples for the product shelf life testing. And let's take a look at a total HIP system, which typically consists of 4 components, like an acetabular cup, acetabular insert, and femoral head and femoral stem. So because each of these components can be manufactured from different materials. For example, acetabular cup and femoral stem are generally manufactured from metals, such as titanium alloy, which is very stable. So product shelf life testing is not needed. However, for both of these 2 components, they could include a surface coating, such as titanium coating and calcium phosphate coating, due to their potential instability. So for calcium phosphate coating, we would like to say our product shelf life testing. And for the femoral head, the metal one is stable, but for some types of ceramics, they have a hydrothermal instability. So we also need to say product shelf life based on this material. And for the acetabular liner, they could be manufactured, most of them manufactured by polyethylene. And for the PMA, like a class 3 ceramic on ceramic total HIP system, they could also include some zirconia. So in both cases, we will, because of their instability, we will need to say the product shelf life testing. So our first example is the polyethylene implant. So FDA has issued a guidance on this material in 2019. So here are some images of this specific acetabular insert. So this guidance recommend the mechanical properties of polyethylene that contains unstable free radicals may degrade if the product is exposed in air during shelf storage. In addition, the shelf life of this material that has been irradiated and packaged in an inert environment may be limited by the integrity of the packaging material. Therefore, we recommend the stability of this material be assessed for the duration of their specified shelf life. And we also recommend to evaluate critical device properties, which are listed in this guidance, mainly the materials and the mechanical testing. And we recommend repeat all this testing. And also we recommend the device and undergo real-time aging to determine definitely the effects of aging on the maintenance of sterility and the device performance. So again, if you use device subjected to accelerated aging, so here are some general language that I mentioned earlier. We would like a rationale why this is applicable to your device. And so while this presentation is focused on the stability of the device, please note that while shelf life has been determined through both packaging integrity and product shelf life testing in pre-market review, non-conformance in the integrity of packaging can significantly affect the device stability and lead to device failure, even if the device is within the labeled shelf life. So FDA recently issued a safety communication on risks with Exactech joint replacement devices with defective packaging to remind patients and healthcare providers about the joint replacement devices manufactured by this company and to their records in the last 2 years. So I provided a link here. You can have further information if you are interested. If you are interested, please check for further information. So all their joint replacement devices contain a polyethylene component, which should be in packaging that contains multiple oxygen barrier layers, as indicated in the package specification. The recalled device were packaged in defective baggers that were missing one of the oxygen barrier layers that protect the device from oxidation. So this oxidation, can degrade the polyethylene over time, and which led to accelerated device wear, failure, and the component cracking or fracture, all leading to corrective revision surgery and even bone loss. So that's the reason these products have to be recalled, due to the packaging issue. And my next example is on surface coating, specifically on the calcium phosphate coating. So again, we asked the company to evaluate the critical physical, chemical, and mechanical properties of this calcium phosphate coating to ensure the coated device will perform adequately and consistently during their entire proposed shelf life. And we also needed the firm to provide the protocols used for the shelf life testing. And for some resorbable calcium phosphate coating, we would the firm should conduct testing on both real-time on real-time aged samples to confirm the results of their accelerated aging study. So this testing again should be conducted in parallel with submission review, with the results just needed to document it to file in their design history file. And next example is resorbable bone void filler devices. And our current recommendation is the shelf life studies should evaluate. I have used this multiple times, critical chemical, physical, and mechanical property of this device. And also data supporting the expiration date for the final finish sterilized, this resorbable bone void filler should be submitted based on real-time stability testing. And the following parameters should be evaluated upon real-time storage over the shelf life of the product and collected from at least 3 production lots. I'm not going to read through all these parameters, but just want to reiterate that these are there are multiple variables which are device specific, and also some language regarding the combination product, because accelerated stability conditions can detrimentally alter the characteristics of resorbable bone void filler devices containing biological derived components. It is necessary to validate any accelerated stability testing with real-time stability testing for the same stability parameters. Once validated, the accelerated stability tests may be used for future stability assessment. And this is our example for patient-matched guides and implants, which are slightly different from other products because they are patient-matched. So for this type of device, shelf life should reflect appropriate duration between the acquisition of patient imaging and the planned surgical intervention to ensure that the anatomical situation has not changed, such as the guide performance can be affected. So the consideration should be taken for the age and the growth rate of the patients. And also the shelf life should be based upon the indicated patient pathology and the sensitivity of the patient-matched regions to continue the disease progression. Also, all patient-matched guides rely upon a specific geometrical configuration to establish a unique alignment onto the patient's anatomy. We also recommend that a guide deformation as a result of shipping be considered. And additional dimensional testing should demonstrate that guides do not deform following simulated distribution testing. As I said, the product shelf life testing is device specific. So for any other device, we would recommend you check with the OHT office that regulates the specific device for relevant guidance documents or standards. For example, for our OHT1 product, they do have a standard which gives a very comprehensive recommendation how to address the shelf life and the transport stability testing for intraocular lenses. And for more specific question, we recommend you to submit a pre-submission to request feedback per the FDA guidance document for Q submission. And I have provided a list of reference, some FDA guidance document you can download from FDA website. As Steve said, for the standard, I guess there's a fee associated with this standard. So finally, I would like to acknowledge all of my colleagues from Office of Orthopedic Device, as well as from other office within OPAC, and also our colleagues from FDA International Office with Patty and Catherine for your invitation and support. Thank you. And my email is listed here. I think Steve just listed also with and Patty's email.
Thank you very much, Liman.
Thank you.
I think that we can move on to the questions that we have already received in the box that we've been monitoring. Thanks to all the participants for including those questions ahead of time. And at this point, I think that's good. We have everyone on camera, and I'll go ahead and just read the questions. And please free to jump in, Liman and Steve. So one of the questions that came in, it says, do you request the test results for residual after EO sterilization, such as ethylene glycol or ethyl chloride? If so, are the analytical standards used to measure these residuals considered medical devices?
Oh, good question. Are the devices used to make the measurements considered medical devices? I don't know if that I would.
So I think there's a two-part question. So one
Yeah.
is, do you request the test results for residuals after EO sterilization?
Okay, yes, that I understand that. I'm not saying yes. I'm saying if you turn to our sterile devices guidance document, the very first item in there under Roman numeral 5 specifies, the information that we want to see for a 510K. And for a 510K, it says, and I'll look at it specifically, for chemical sterilants, so it's not just for ethylene oxide, the maximum levels of sterilant residuals that remain on the device, and an explanation as to why those are acceptable. So in other words, we're not necessarily looking for the test data in a 510K submission. We just want to know what the limits are that you intend to allow that device to meet. So that's what we're looking for a 510K. Incidentally, it also applies for other chemical sterilants, for whatever is whatever the sterilant is being used. For other devices, and in certain circumstances for 510K, we may ask for it, but for other devices that are PMAs, class 3 devices, we want to see all of the data, all of the test data. And for that, I would refer you to 1099-3-7, ISO 1099-3-7 specifies what we're looking for. We're looking for ethylene oxide residuals and ethylene chlorhydrin residuals, but ethylene glycol residuals have been dropped out of that standard, so they're not included there. But it does give you, the information that we're looking for, for depending on what the type of device, what type of intended use and patient contact, that'll give you what the limits are. And what I've said before is that the testing has to be done. The data does not necessarily have to be provided with a 510K, just what the limits are, but the testing does have to be completed.
Thank you, Steve. And I think the second part of the question, I'll read it, but I do believe that we may need a little bit more clarification because this question, it's not very clear, I don't think. So it says, if so, are the analytical standards used to measure these residuals considered medical devices? So are the standards considered medical devices? I would say no, but if there's any clarification that you would like to add, I think we can get to it maybe later.
Yeah, and I would think that the standards are not considered medical
Correct.
devices. The equipment used for it, I don't know the answer to that, but that could be like an analytical, like all the other types of testing and measurement devices that are being used. I don't think that they are, but that's not my area of expertise.
That would be correct. Sorry, Lemon, did you want to add anything?
No, I
Okay.
don't have anything to add.
Okay.
This,
But I mean, I would go back to the definition of a medical device, which is a device, a product used to treat a particular condition or a disease. And if those particular equipment are not used for that purpose, then it would not meet the definition of medical device. So I would just indicate that we would look at the definition of medical device for that purpose. Going to the next question is, how often the analysis of the tests conducted in accelerated stability and real-time studies?
Could you repeat that, please?
How often the analysis of the tests are conducted in accelerated stability and real-time studies?
Okay, the way I'm going to interpret this is that for certain testing of processes in general, sometimes they are test it once, and some of them say you have to revalidate this process on a regular basis, like radiation, sterilization. You have to revalidate it or do a dose audit on a regularly scheduled basis. For packaging, well, I would refer to the standards for these, but for packaging, I don't think there's any routine revalidation, but in the guidance document that I referred to, and you could see it in one of the title in the table of contents for 16775 is change control. So sometimes there's subtle little changes that are done, maybe to the device, maybe to the process, and that'll give you an opportunity, access to guidance on how to evaluate whether you need to revalidate something that's under the category of change control.
Thank you, Steve.
Yeah, for the device performance testing, I think you generally just, if you are using real-time aging testing, you may want just to do different time points. If you already have the accelerated, aging testing to define your shelf life, you may want to continue with the real time, just to do different point until your level the shelf life based on that accelerated aging results, to make sure it can be the same. So.
Thank you, Lemon. Next question says, do the labeling system, packaging, shippers, corrugated boxes, IFUs need separate agent tests, and would that be recommended?
The shippers and the
Boxes.
boxes. No, we don't for aging, for stability, no, that should be part of the performance testing, however it is shipped. But there's no, our ultimate concern is maintenance of sterility and how that is validated. So that really needs to be, the shipping containers are a separate issue, and there's no need for aging simulations or real time to validate them, because there's really, when it comes to the accelerated and real-time aging, we're only looking at seal strength afterwards, and there's no seal strength involved with the boxes or the pack or the shippers and things like that.
Okay. So I think that clarifies it, because it says, would you recommend it? I think we would not require it. I mean, we could say that we would not require it for that, for packaging. Are the manufacturers allowed to modify the shelf life after the product is on the market? And how is the maintenance of the SBS demonstrated when sampling products at the point of sale?
Okay, for the first question, first, I want to make a general disclaimer. I'm giving general answers here, but sometimes, if there's a real particular situation that's a little nuanced, that I'm not getting all the information on, it's hard for me to address that very specifically. So I'm responding in general terms. I just want to make that clear, that I'm not giving you specific recommendations for your particular case. You can come in and do a pre-submission for that, or just call up DICE and get some information. So I'm sorry, what was the first question?
Are the manufacturers allowed to modify the shelf life after the product is on the market?
Yes, absolutely. The regulatory pathways for that differ a little bit. I think for a 510K, it's called an add to file, I think, but you need to confirm that with a regulatory specialist. But yes, you can extend the shelf life. And in fact, that's not uncommon that somebody wants to get on the market and they can establish a 1 or a 2 year shelf life. And then they do the additional testing to extend their shelf life. As long as the testing's done, that would support it. And in fact, I'm talking about 510Ks, class 2 devices. For class 3 devices, PMAs, it was a few years ago. I don't know if the PMA approval letter has changed, but there used to be a sentence in there stipulating exactly that if your validation protocols have not changed, if you're doing the same testing, but you're extending the shelf life, you're just generating some more data using all of the testing that protocols that you've already used and we've already evaluated, then you can do that.
Yes, and I think Lemon in her presentation indicated that.
Okay.
Yeah, I have a slide, I think 12, talked about different scenarios for 510K and the PMA. And yeah, it's very common, company extended their shelf life due to various situations. One situation could be, that's based on the real-time aging, and initially it's very short, and they can do additional testing. And once that's ready, and they can ask for, they actually, for 510K, they even don't need to submit a 510K, new 510K if they're using the same method than used in the original 510K. So there are different situations, but as Steve just mentioned, for PMA, if they are using the same method already approved, they can just report that in the annual PMA report. But if they are using a new protocol, they needed to submit, a PMA supplement to get approval before they can make that change, the shelf life change, anything. So just want to say it's very common. Yes, they will extend their shelf life due to different reasons.
And just to clarify, for the attendees, changes shelf life as long as it's within that, we've reviewed the protocol. As long as they're doing testing according to the protocol, we would not see a new submission come in for that, right? They just need to file it within their records.
Yeah,
that-
basically for 510K, yes, they can do
510K.
that. And for PMA, they needed to include that in the annual report. We
Correct.
don't need to see the test report either. So
Was there a second part to that question?
How is the maintenance of the SBS demonstrated when sampling products at the point of sale? I'm not clear on this question. Does that make sense to you?
Could you repeat it?
How is the maintenance of the SBS demonstrated when sampling products at the point of sale?
At the point of
If
sale.
if I can elaborate a little bit, is what their question is, when you sample a product on the point of sale, what are the tests or that have to be conducted, or how they can demonstrate that the sterile barrier is still intact?
Okay. Good question. There's actually no what we call test for sterility of the product. I mean, yeah, there's no test for sterility. You don't open the packaging. You don't aseptically evaluate product in a statistically meaningful number to demonstrate that it's sterile. Instead, it's been demonstrated over a long period of time that some of these other tests are more effective and more valuable, and you don't have, with that type of test, a test for sterility of the product, you can get false positives and contamination. But if you do the other testing that was mentioned, for example, dye penetration, or they have bubble emission tests to find leaks, or I have not seen this in quite a while, and I don't think there's a standard for it, but there used to be something called a microbial chamber challenge test, where the packaged product would be put into a chamber with an aerosolized microbial cloud and challenged, and then it would be tested for sterility afterwards. The package would be aseptically opened and see if it's been contaminated. But the problem with doing what is suggested, doing a test for sterility of the product, is that there's no real challenge to it. There's no pressure trying to push microbes into the package. There's nothing, challenging the package. So if you were to test 30 packaged products, unless something traumatic happened to the packaging, it's likely just sitting there and no microbes are getting in. So it's not a very meaningful test compared to something where there's a real challenge, like trying to push dye into it or something like that.
And then Steve, it says at the point of sale, so we wouldn't, if they wouldn't require tests at the point of sale, can you, for sterilization, for example, right? Like after.
Yeah, I think all of these tests are really done on final finished product before it leaves the
Before it leaves the
commercial.
facility
the facility.
for sale.
Yeah.
Okay, I think we're ready for the next question. Unless Lemon, did you have anything to add to that one?
no, I
Okay.
don't have.
How does the FDA verify that the test results submitted by the manufacturer were conducted using validated methods or techniques, and the sampling supports the extrapolation from batch to the whole production? And the second part of the question is, and how does the FDA authorize the laboratories that conduct these tests in the US?
Lemon, do you want to go first?
Yeah, so start from the second part. So I think currently we don't really have any lab that are authorized by FDA. It's best basically testing lab like used by the firms themselves. So, but we, for PMA, we do need to see all the testing report as we mentioned earlier. So to make sure they are following, the test review, they are following exactly what we are looking for. But for 510K, so we do have some standards. So they, with regards, I know Steve, you have a lot of standards. So you may need to see if they follow all those standards, but for device specific performance testing, basically they are just repeating all those testing. So they did initially when the product was just manufactured. So they will repeating all those testing. Those testing lab, so if they tell us they are doing exactly the same thing, they are following exactly the same thing. So we generally accept what they said because it's the same as when we reviewed their original testing data. So just want to hope that answers this question.
And from a microbiology perspective, I guess there are a couple different things that are involved there. You could be talking about packaging validation or industrial sterilization or reprocessing instructions. Whenever we see, whenever we need test data, a 510K, we'll spell out, we want to see that it's been done, and it'll be looked at upon inspection. And for a PMA class 3, we're going to want to see all the data upon submission, and we want to see all the details. So we want to see the validation, the protocols, the report, and all of the things that are involved with the validation to make sure, we'll check and see that you have proper controls, and what your acceptance criteria are and that they're justified. And when it comes to package stability, for example, you should have your accelerated aging calculations documented. And again, for 510K, it'll be looked upon inspection, but for a PMA submission, we'll look at it upon submission, at the time of submission.
Thank you. And I mean, I think there's standards out there, right, that have protocols on how to conduct some of these tests. I think that you have already covered.
Yes, yeah, those are great guidance, good recommendations, good requirements when you follow them.
What is the applicable stability standard in the case of gel devices, and such as the ones used for ultrasound? I know that you don't have you cover majority of the experience in orthopedics. I'm not sure if you know, but if not, I think we can take this question and respond later. But do you happen to know, Steve?
For gel, is that what you're saying?
For gel devices, such as like ultrasound gel.
I don't have experience with that, but I do know this is a one-off that when we drafted a guidance document for antimicrobial coatings, for that a decision was made not to accept accelerated aging for that, because that Q factor could vary too much. So I don't know the answer to this, but I'm guessing that they have a very solid reasoning for whatever they recommend, and I would suggest reaching out to them in particular, that particular office.
Yeah, so I think this would be a separate office within FDA, just to clarify. So I think what we could do is after this particular webinar, we can follow up with this particular question and provide any standards for gel devices coming out of that office. So
and there may be a dedicated guidance document for those devices.
Yes.
In particular. That's a type of example where it's a little bit unique, and they may have very unique specifications and recommendations. So it's worth looking up.
Correct. So we'll follow up on this particular question. The other question is, do the devices that incorporate human material have to validate the sterilization process, and what would be the most frequent method used? For the use life, does the FDA require specific testing, or the manufacturer could choose the ones to use? Again, I think this talks about incorporation of human material. I'll leave that up to you if you can answer. If not, I think this is another one that we could take back and answer at a later time.
Lemon, do you have anything you'd like to comment on that?
I think they are asking for specific sterilization method,
Oh,
right?
okay.
It's
Incorporating, so devices incorporating human material.
Yeah.
Do they have to be validated? Do they have to have a validated sterilization process?
Yes, they do. If there anything that comes with a label sterile has to have a validated sterilization process established for it. Because I'm in orthopedics, I've not seen any of those. I've not reviewed those myself, but a colleague of mine who has done some consulting with biologics, I know that there's I'm almost certain that there's a FDA guidance for that. And also, it's either an AAMI or maybe AAMI ISO standard for devices that incorporate biologic products. Yeah, but not my area of experience.
Yeah, so I think this is another one that we could provide some guidance at later.
I did have a slide talking about resorbable bone wall filler, which may incorporate some biologics. So, but that one is not about sterilization. It's about device specific testing. You have to consider those biologics if you are using accelerated agent, because usually you may not have a very well established accelerated agent protocol for that. So that needed to be very careful.
That's a good point.
And another kind of minor note is that for those device types, for those components, they may be more vulnerable to sterilization methods than metallic or polymeric materials, of course. So there are ways of approaching that. If you want to get a sterile label on something, demonstrate an SAL of 10 to the -6. There are methods available that are called, just to make it easy in terms of steam and ethylene oxide, there's an overkill method of sterilization, but then there's something called a BI, biological indicator combination, BI bioburden method, which allows a reduced extent of treatment and still provides an SAL of 10 to the -6.
Okay. The other question, there's another question that says, can you clarify again what the difference between useful life and shelf life is?
I think that's
I think you had a definition.
yes. So shelf life, I think is let me double check. So shelf life again is after that expiration date, right? You cannot use this device anymore. But useful life is once it's been used in the let me say to make sure I'm following exactly. So the duration of actual use, for like a lot of, I'm also talking from orthopedic, space. For example, some of the polyethylene products in the body, they will not be able to, after a certain time they wear off, they will lead to failure, then they will not be used as long as, your lifelong. That after certain years, it will, too much wear and it will not function well. So that's in general the useful life. Like we talk about hip implants, maybe their useful life is generally 15 to 25 years long. So that's what we talk about, shelf life. Useful life. but also Steve also talked about some are reusable. So they are for reusable device, they are also number how many number of repeat use before they, they cannot function as intended. So I think Steve, you also showed like some pitting, if you see those things happen, then you're not supposed to use it anymore. So that's how many times you already used. That's number of use. That's also for useful life. So
Yeah, I think I'm confirming with what you're saying, but I know reusable medical devices, for those there is a use life or a reuse life, which is the number of times it can be used before it starts to degrade, or a certain number of times that it's been validated for function, good performance and functionality. And for shelf life, I think sometimes this is used, the expression is used a little bit in a mixed kind of way, but when I see it on packaging, I see it for shelf life or expiration date. I'm just looking at it from a microbiological perspective, but that is for a terminally sterilized product, how long it can be on the shelf before it should be used. There's a little bit of overlap, or a little bit of mixed use, I think, of the terminology. I don't know. I can check while we're here to see if there's an AAMI definition for these. And we can come back to it.
Sounds good. Most part of orthopedic implants are non-sterile and undetermined validity. In this case, what do you need to provide?
I'm sorry, most of the orthopedic are non-sterile.
Are non-sterile and undetermined validity.
I don't think most of orthopedic devices are non-sterile. The majority of them are provided sterile. So
but
because
let's say if they're labeled non-sterile, let's say, although we know a lot of the implants that we see are going to be
sterilized,
sterile, right?
Yeah,
sterilized and labeled as such. But I think the question, it says if they're non-sterile, what do we need to provide? I mean, it depends on what the device is, I guess. But
so basically, they needed to. I will start, and Steve can add. They needed to provide a validated, sterilization cycles. For most of them, I think is steam sterilization cycles in their labeling, so that the end user, the hospital, they can do those. Because most of the facility may not have gamma radiation or other type of device, but they do have some steam sterilization facility. So I think, but they needed to make sure that's validated. They needed to let the FDA know that. So.
Yeah, for reusable devices, there should be validated cleaning and sterilization instructions. And in the cleaning instructions there, again, you can use that guidance document, but you're going to have basic information in there like point of use treatment, visual inspection for cleanliness, an instruction to thoroughly clean, final rinse water quality that's supposed to be used. But then when you get to the sterilization part, there should be, there's typically a table that includes the time for steam, time, temperature, and drying time, and recommendation to use an FDA cleared sterilization wrap. If you've got a situation there where there may be confusion, it's important to let the end user know that the device is not sterile already. So, if it's packaged in a way that makes it look like it might be sterile, it's important. And again, I refer you to 11607. They've got some good graphics in there to ways to indicate that this product is not sterile, or the product is sterile, that it's got a sterile barrier system around it. But if it's clearly indicated non-sterile, it's a reusable device, those are the things that have to be included in the labeling. And sometimes it might be a single-use device that is for a procedure. It's not implanted, but it still has to be sterilized by the end user, or maybe cleaned and sterilized by the end user. whatever it is that you determined based on the intended use, you have to have adequate labeling to account for all of those things.
And Steve, can you clarify in that situation, we would require to review a validated cleaning and also sterilization process, like a protocol for that, right? For the end user?
That's correct. There should be,
Yep.
The labeling should accommodate everything, but all of the validations should have already been completed. And we're very fortunate. We just published, Amy just published in the end of 2022, Amy ST 98 on requirements for validating cleaning of medical devices. Before that, there was TIR 30, which a lot of people knew about. It was just a TIR, it was technical information report. This is a first standard, comprehensive standard for cleaning. It's available now. And I would recommend referring to that for all of the procedures, all the test types for cleaning and the types of controls. It goes into the controls really well. And then when it comes to the sterilization, you can refer to the standards that have been out there for a while for steam sterilization and also for ethylene oxide. Yeah, so everything has to be validated, even the drying time. So if you're doing a steam sterilization, we want to know that the drying time that's recommended in the labeling has been adequately validated.
Right. Are the stability studies for medical devices required to consider climatic zones? So different climate zones.
For packaging, are you talking about packaging? I'll turn it over to you in a second, Lyman. For packaging, part of that chart that's in ASTM D4169, there's that big table 1. It has 18 different distribution cycles and all of the different assurance levels. There are specifications in there for transportation through climactic changes. So you can refer to that and it'll give you ideas about pressure changes and humidity and things like that. Depends. All of those different options, that 1 through 18, they're all dependent upon how you intend to ship it. If you're going to, just ship it across town, the demands may not be that great. If you're going to ship it across, several countries and over oceans, the demands are going to be greater and you have to validate it using that way. Or all the way in the back of that standard, it gives you the option, if you're just designing and making an instrument within a facility and you're just shipping it from one end of a hospital to another, to the building next door or something, that allows you to, go with different things. But anyway, all of those things are different variables, including climactic conditions that are addressed in that particular standard, the ASTM D4169.
Thank you. I'm being cognizant of time here. So we'll take a couple more questions and maybe the rest we can take them back and provide as a written response.
Okay. Incidentally, they just made a change to the accelerated, I think it's the accelerated aging standard that now includes humidity.
Includes humidity.
And there's going to be a slow, we have a transition period. If you've already done this data, or if you're about to start testing, you should be using the new one. But if you've already tested it, there's a grandfather period. But I think I've not read it myself yet, but I heard that the new accelerated aging, I think that's the one, has a humidity consideration in it now.
And then the other question is, are these required for investigational devices? Like all these information for IVDs?
I think these are factors for consideration during the IDE investigational device review. So we will let the firm know you have to consider all these things to make sure. But if it affects safety, so it may be big concern. So, but we will let the sponsor know you need to consider this, but all this information need to submit it during your PMA or 510K submission at that time. So may not, yeah, it's just a factors we'll consider. I don't think we will need to review all the testing reports for this yet, because yeah.
For what we do for microbiology, packaging and sterilization and packaging is the first thing when an IDE comes in, the first question we ask is, what is this device going to become, a 510K or a PMA? If it's going to become a 510K, we want to know, all of the testing that has to be done. We want to know that it's been done. If it's, but we don't need the data. So we just need to know that it's been done, just like as we were about to accept a 510K. But for a PMA, we do want to see the data for sterilization and packaging and everything else. We want to know it's been done. Sometimes we can be a little bit flexible in some areas, but the bottom line is patient safety. So everything that's required for patient safety for a 510K, we're not going to go any more in depth for a IDE that's going to become a 510K for sterilization and packaging. But for an ID, because we'll expect to see that once it's inspected. But for something that an IDE that's going to become a PMA, we're going to ask for all the data that is reflective of our concerns for patient safety.
Very good.
And
Thank you.
Incidentally, for some of those things, there are qualified shortcuts. So in other words, in 2 of these sterilization standards, you can do single batch lot release for sterilization instead of doing a huge comprehensive sterilization validation. There are sections in those FDA recognized standards that allow for single lot release. Yeah.
Sounds good. I think we have about maybe 5 questions or so left that we are not able to cover just because due of time. I just want to be cognizant of that, but we will do our best to provide those as a written response after the webinar, just so to make sure that we're not leaving any questions unanswered. With that, I believe I see Teresa maybe wanting to make some comments, and I think after that we can go ahead and conclude the webinar. Teresa, go ahead.
Hola, pues, gracias por el espacio.
Thank you for the opportunity. Thank you, Nini and Steven. Both of your presentations were excellent. I would like to say that regarding the status of standards in Mexico for stabilities on medical devices, it's a great pleasure to tell you that we have published a supplement for medical devices 5.0 of the Mexican Pharmacopedia. This is what helps COFEPRIS Appendix 9, which is called Stability Studies for Medical Devices, and it is regulatory, will be enforced June 9, 2023. According to good regulatory practices, the Medical Devices Committee has taken the guides that the FDA mentioned so as to harmonize our standards and international standards so the conditions of those studies are the same all throughout the region. In the past, we had attempted to do this work, but it hadn't been possible. Medical devices, it's a huge world. And so it wasn't until now that we were able to create guidelines considering the guidelines that were mentioned, the ones that were quoted, ACTM, FN 1980, and all. Thank you very much for sharing your knowledge, both of you. Thank you.
Thank you very much, Teresa. Thank you, Lyman and Steve, for the wonderful presentations as well. There were some really good questions, and thank you for all the attendees for those questions, because it really allowed for some really good discussion after the presentations. Just to summarize, as we heard earlier, the stability studies for medical devices are crucial in pre-market review to ensure quality, safety, and effectiveness of these products. And Steve and Lyman really provided a great overview of what FDA requires, the guidance documents that are available, and the standards that the agency recognizes for tests that are accepted by FDA. So we do encourage that the attendees to look at these guidance documents that we shared and the standards to get more in depth and look at how these tests are conducted or how FDA what FDA requires. Again, I think by collaborating together, we hope that we can learn from one another and align these requirements to streamline these processes, to still have oversight and assurance of safety and effectiveness over these products. As regulators, that is a requirement of ours, but really while still removing some of the barriers and trying to create ways to bring products to market quicker for patients in our countries. Again, I just want to thank everyone for your participation. And at this point, I'll just hand it over to Sandra to see if she has any other last minute words before we conclude the webinar.
Thank you very much, Reza, and really thank you very much, Stephen and Lemon. I think it has been really, really as helpful as we thought to have this session. There are still many questions open, and we're very appreciative for you taking the time to respond to as many as we could under the time available. And of course, we are here very enthusiastic to continue to learn more and more about this very, very relevant topic for our sector. So thank you very much. And thank you very much for a very active participation of all of our attendees during this session. Besa, Patty, and all the FDA team, thank you very much again for all your support for the activities that we are organizing under the MDRC project that intends to help all the relevant stakeholders to get a common understanding on critical topics that will allow us to further advance the regulatory convergence that is really most needed in our sector. So thank you very much again, and enjoy the rest of your days.
Thank you. I would say one more thing. I think we're looking for feedback. And so for those that, I think we covered a lot during this webinar, but we're always looking for feedback to see where, what other clarification and maybe what other else is needed in this topic to align those regulations and really increase confidence in utilization and taking similar approaches. So I think Steve and Lemon already shared, my colleagues Patty Pineda's email and mine as well. So we would appreciate any feedback that you have about this webinar. We take those very seriously to bring together these courses for everyone. So please feel free to use those. And with that, I think we're good and we can conclude the webinar. Thanks again for everyone that joined.
Thank you.
Bye. Thank you, everybody.